All right, so we're going to start talking about transcription today. So we've talked about DNA replication, we've talked about DNA repair, right? Um, and so we're going to start talking about transcription. So when we think about this, central dogma of molecular biology, which is DNA goes to RNA. You have a DNA, which is a perfect copy. You go to RNA, or we hope it's a perfect copy. It goes to RNA, which is some photocopies of that perfect copy, and then use those instructions in that photocopy to make proteins, which are the functional units, functional or structural units, usually. Sometimes it's RNA. And so we've already talked. So we've talked about this process here, right? Actually, I'm skipping part of replication because it didn't show up in the notes, so I'll have to bring that back. But we've mostly talked about this, okay? What we're going to need to do is go through replication and the three parts of it, one of which we talked about for that mismatch repair that are important for the fidelity of replication. Okay. So, but we'll come back to that. So today what we're going to be talking about is this bit, okay? Most of what we talk about, yeah, it depends on how far we make it, but most of what we'll talk about is that part, which is transcription. We also have introduced and talked about, well, sorry, not that. We've talked about reverse transcription, right? Which is where it's what our topoisomerases are doing. We're going to see that when we talk about viruses, so we will talk about reverse transcriptase specifically in like a viral context. We've talked about that as well. So this is an example, and what we're talking about is most of our examples are going to be bacterial. They're a little simpler, they are better studied, so most of what we talk about is bacterial. So here this is an example or a cartoon of a bacterial transcriptase. Okay, and so what this is going to do, this is RNA transcriptase, and so what we're going to do here is when we think about the mechanism for this enzyme, it's going to have three bits to it. We talk about, you're going to see this again and again, all these enzymes, you kind of think about this. You have initiation, okay, you have what's called propagation, or let's say elongation, okay, and then we have termination. And they mean just kind of what they say. So initiation is the enzyme or process gets started. This is a single enzyme, so we can just say enzyme. And specific things happen there. Elongation is going to be kind of the fast, it's going to be the fast transcription. That's very processive. We'll talk about that in a minute. What does it mean when we say processive? That's how many it can add before it falls off. Okay, so you've got the transcriptase that's sitting on a DNA template. And then termination is your enzyme's going to stop and it's going to fall off the template. Okay, in all cases here, we're talking about transcription. Our template is going to be DNA. You guys know this is DNA, and we're saying the word transcription, we mean RNA is made. Questions generally? Okay, this is the mechanism of adding a base. This is the same as the mechanism, so it's a more complicated picture with magnesium. You don't need to worry about this part, like what the enzyme active site is. Although you'll notice what we've got, we saw some of this, some of you did mutants in lab that match this, right? What amino acids do we have holding these magnesiums in place? Aspartates, right? Aspartate 124 and 154, the MTHFS, are coordinated with a magnesium ion, which then coordinated with the phosphate backbone. And in this case, it's the phosphates on ATP to hold it in place. So some of you did that where you mutated the aspartate, so presumably your hypothesis would be that if I mutate the aspartate to an alanine, then the magnesiums aren't held in place and they're not going to hold the ATP in place. So you don't need to worry about that. What you need to be able to do is if you're given a strand like this, you need to be able to show the mechanism of it reacting. So here we have a triphosphate reactor with a triphosphate, and if we're going to have pyrophosphate come out, what happens to that pyrophosphate immediately? Right, it's going to be, we've got an enzyme that's going to go ahead and take it to two phosphates, right? And that's a driving force. It's a coupled reaction with this. So we get the energy from this reaction and plus the energy from this reaction. Questions? It could be the same diagram I showed you before, which maybe was a little less complicated. The only difference when it's RNA is what?
Uracil.
Well, thymine and uracil, but the bigger difference is if it's DNA, this OH isn't here, right? And if it's RNA, it is. All right, RNA polymerases. So DNA polymerase can start at any 3' end, right? And we've seen that in our repair mechanisms, right? We get a nick in the DNA or something, and DNA polymerase can come in and start on that end and get rolling, right? Where we have a big section that's removed. So DNA polymerase can start at any 3' end. So if DNA polymerase falls off, let's say it's not very processive, like DNA polymerase 1 is not very processive. What's the consequence? What happens if it falls off?
It stops and we feel like adding the primers.
Okay, so let's say we've got DNA polymerase going this way, right? We've got a 5' end, a 3' end on our template. I'm going to make it the same color so we know what it is. So DNA polymerase is now making a copy of this template. All right, so it's on the top just as I draw it here. DNA polymerase stops. Let's say it just falls off there. It's DNA polymerase 1. It doesn't have what it needs to do, however many that is. Let's say it's 100. It's just too much. What can happen now? Are we done? Do we have damage? What happens next? What was that?
It like does it again?
Yeah, it does it again. It can start at any 3' end, right? So if DNA polymerase falls off, it can just come back down, bind here again, and just keep going until it's all done. Oops, that's a 3' end. Does that make sense? Oh, okay, so that's DNA polymerase. RNA polymerase starts at what's called a promoter. Okay, let's actually add this up here. If DNA polymerase falls off, it can bind and start again at the exposed 3' end. All right, so but RNA polymerase is different. RNA polymerase starts at a promoter, which is a defined DNA sequence. It's a special DNA sequence. Okay, I'm just going to say a special DNA sequence. I guess I could say a specific DNA sequence. Let's say that instead. All right, and that's the only place it can start. There may be several different sequences, but they're not common. They're not flying. They're at the beginning of genes. They're not in the middle of genes. Okay, things like that. So what happens if RNA polymerase falls off early?
You're done.
It's done. Has to go and start over. That's the only thing it can do, right? So if RNA polymerase falls off the template, it has to start over. And it just made a transcript that was not the full gene. All right, so it's made an aborted product. If that thing gets transcribed, and it could, it's just missing the end, it's missing the stop sequence, you could make a partial protein that could fold weird, and if it folds weird, it could have hydrophobic stuff on the outside. You could get a bunch of stuff precipitating, which is really bad for a cell. It's bad when proteins glom together and precipitate out in cells. A lot of these dementia-related diseases have that going on. Okay, so to prevent this, RNA polymerase is very processive. And the lengths of genes are limited. Right? So also you could think about error rates of RNA polymerase. If RNA polymerase makes some errors and makes a bad transcript, if it's a shorter piece, you didn't waste as much. Anybody know what the biggest gene in the human genome is? Biggest single protein unit? It's something called titinin, and it's in the involved in muscle tissue. I don't remember what it does, but usually what happens is instead of making a 10,000 amino acid titinin, which is about how big it is, you'd make a bunch of 1,000 amino acid pieces or a bunch of 100 amino acid pieces that would come together in a complex. So we see these complexes instead. All right, these are some examples of bacterial promoters here. And when we think about these bacterial promoters, there's a term I want you to know. Well, let's just look at these first. So what we have is we have a bunch of bacterial promoters that are going to be, they're kind of all in the same group. They have this region, so we have the RNA starts here, and then there's some space, and that space can be a little bit different. And then we have this minus 10-ish, 10 region, minus 10-ish region, because this space is different. Where you're going to end up what's called a Tata box. So T-A-T-A-A-T, call that a Tata box. But it isn't always Tata, right? We've got Tata, and then we've got Ta, and some other things, right? Okay, tact. But we've got some other sequences that can happen, and RNA polymerase can bind to those as well. They're similar enough. It's not going to bind to all of these with the same affinity. Let's say RNA polymerase binds to a given sequence with a stronger affinity. What do you think would happen with that? So if we were to say when RNA polymerase binds to a sequence tightly, what do you think happens with that gene?
More of it's made.
More of it's made, yeah. More of that, more of the, we're going to say mRNA, we'll define mRNA later, but I'm sure you've heard of it. More of the mRNA for that specific gene horrible time spelling today, is transcribed. Okay, so whether we get more or less of something is dependent on how tightly we get that RNA polymerase bound, how much of it we end up seeing. And so we've got these tanoboxidant different sequences with different affinity. Some we got this -35 region where we have something else that's going to kind of help bind. And if you look at, we look at this and we'll see more of these sorts of sequences later for we're going to look at the lac operon in detail because that was the first and it's a really well studied system. It's also the one that you used in lab in terms of inducing your protein expression. That you can have these other elements that are a ways away, and these aren't even very far away for that can actually interact with this. You can have things that are hundreds or even 1,000 base pairs away that can kind of come around, wrap around, and help with transcription, getting transcription started. So initiation. So these can be different, and then we can also have even more things that are going to be related to helping or preventing that RNA polymerase from binding. But you'll notice that a bunch of those are different. We don't need to see all of them at the same time. The consensus sequence, you could think of it in your head as an average of all the sequences. What it is it's just the sequence that has the most common base at each position. So what that means is your consensus sequence might not even be a natural sequence. It's not the most common natural sequence. We take T's the most common, right? Let's take these 1, 2, 3, 4, 5 that we can see. Well, mostly we see T in the first position, right? Mostly we see A in the second position. Mostly we see T in the third position, so TAT. Mostly in the fourth position we see A from these 5. Mostly in the next position we see, you see what I'm doing here? Looks like an A, is that right? There's only 2 A's and that's it. And then mostly we see T. So it actually matches this one, but it wouldn't have to. You'd go 1, 1, 1, 1, 1. Does that make sense? Mm-hmm. But if you just think about it as an average in your head, that's okay too. Not exactly, not as correct, but I'll accept it. So that's what happens when we think about the consensus sequence for a given promoter. So you see we have lots of different sequences there.
I like this sequence.
Does it have it? Where does it have that? Oh, so it was the consensus sequence. I made it from the 5 lower ones. Okay. And it did actually from this one right here, it actually, the RRNBP1, whatever that gene is, actually did match the consensus sequence in this case. Questions? Okay. Now let's go ahead and run through these parts of the process. First thing I want to talk about is initiation. So they've got binding and initiation separate in this figure. We're just going to call it an initiation. Okay. So if we talk about initiation in transcription. The first part here is the RNA polymerase is going to bind to the consensus sequence. Now you'll notice there's something here that's, we're not seeing just the RNA polymerase. We're also seeing this other thing here. This is called a sigma factor right here. See a little sigma on it? So we have sigma factor there. Before it binds, the RNA polymerase is bound by a sigma factor.
So it's like step 1 and then step 0.5 before it.
Yeah, it's like that. I'm not going to worry so much about if you can tell me what happens. We could put those in a better order here, right? Let's just do that. That may be less confusing. I don't care so much about the order here, but so I do care that you know what this sigma factor does. So it's bound by a sigma factor. That sigma factor is going to cause a shape change in the RNA polymerase. Notice that the sigma factor isn't binding with the sequence. It's going to cause a shape change in this and direct it to a family of promoters. All right, so if we think about RNA polymerase in bacteria, there isn't an RNA polymerase for one family of promoters and an RNA polymerase for a different family of promoters and an RNA polymerase for a third family of promoters. There is one RNA polymerase, and its shape is modified by whatever sigma factor is binding to it. And you could imagine in a bacteria, depending on what it's doing, you'd want a certain set of genes expressed, like a big group, okay? So a good example of this is when you heat shock. So you guys heat shocked your bacteria to get them to take up DNA. That heat shock was at 42 degrees Celsius, if you remember correctly. So what we have is, so sigma factor 70, and the sigma, the 70 refers to a sedimentation coefficient. So it's like a molecular weight, sort of. I don't know if you guys probably, in biology, you learn about sedimentation coefficients. You centrifuge them, you kind of see how, you see where, in a gradient, you see where they end up, essentially, in that gradient. So it's not exactly a molecular weight, but it's kind of close to one. So sigma factor 70 is for, let's say, normal gene expression, or for gene expression in normal conditions. Okay. If the bacteria is exposed to like high temperature, 42 degrees, let's say, if it's exposed to higher temperatures, then sigma factor 32, yeah, 32 is going to be made. And that will cause a shape change and a different set of genes will be expressed. The shape change is in the RNA polymerase, so it recognizes the different sequence. Okay, does that make sense? And that doesn't mean that all of sigma factor 70 goes away and it's replaced by 32, but you start expressing some 32 and you get some more of these genes. What would a bacteria maybe need to do if suddenly it got hotter?
Like,
Might need different enzymes, more specific?
like cholesterol and the membranes.
Yes. Yeah, the membranes are going to have to change, right? If it gets hotter, you don't want those membranes have a certain amount of temperature that causes them to be the right amount of fluid and not fluid, right? How stiff and how fluid they are. If you make it hotter, they're going to become more fluid, and so to get them back to where they're stiff enough to protect the cell, but they still move, you might stick more cholesterol in depending on their contents, right? Cholesterol, you either take it out or put it in depending on the context. It's complicated. But certainly you might make longer fatty acids right, for your membrane lipids, so that they were stiffer because the temperature is now higher. And so you'd expect the opposite if you put them in the freezer, right? We take our bacteria, put them in, let's say put them in the fridge, that they might start making some smaller ones. Okay. Questions? Or if you were to take your plates and you'd done your plating and you weren't going to be able to come in the next day, I stick them in a drawer and let them go over the weekend. And I generally get about the same result that you get the next day. Okay. But if I stuck them in a drawer over the weekend, they would make shorter fatty acids than at 37 degrees because it's substantially cooler. Does that make sense? They're shorter, so the membranes become more fluid in the lower temperatures to make them the right fluidity for those temperatures. Questions on sigma factor? Okay. So then what's going to happen is the DNA is going to get unwound. Okay, we get this unwinding. So we've got some sort of helicase activity here. The DNA template is unwound. And then we have an 8 nucleotide sort of primer or starter RNA is made. Okay, this is complementary to the sequence. It's complementary to the template sequence. So it's not some random thing or made off of some other template. This is sometimes made many times. It's probably most often made many times. So in other words, what goes on is this thing starts and it goes, okay, it moves kind of forward and makes an 8-mer and that falls off, and it makes another 8-mer and that falls off, and it makes another 8-mer and that falls off. You get a bunch of them made, and then finally you manage to get, talk about promoter clearance here, but finally what's going to happen is it gets it so it's good enough, whatever defines that, and the sigma factor will release is the final bit here, and then you're in the promoter phase, or the elongation phase. All right, questions? All right, so then we can look at this. Actually, we've got our elongation phase right here. Okay, so in the elongation phase, this one's not going to be so interesting. The RNA polymerase is going to very quickly and very processively, it's not going to fall off anywhere in there, make an RNA transcript that is complementary to the DNA template. Okay, questions? When we think about this process, okay, or even the previous process, there's a couple terms I want to talk about. So when we do this, this is this strand right here, this top strand, the one that is not the template. This is called the coding strand of the DNA. Okay, and it has the same sequence as the mRNA that's being made. And then down here, this is the non-coding strand. Okay, also called the template strand sometimes. Okay, and it is complementary, it's the complementary strand to the template that the RNA is being made. So if somebody says, or if I have a test, coding strand, non-coding strand, template strand, I expect you to know what we're talking about there. I might ask a question where I ask you to give me the sequence of something made from this DNA and identify what's the coding strand and what's the non-coding strand. And if I flip it upside down, hopefully don't be confused. Okay, then finally we have termination. More complicated than what we see here sometimes, but the RNA polymerase pauses, and this is going to be sometimes due to a structure like one of these. You see this hairpin that's formed here? And it's going to pause due to a termination sequence. And then it falls off the template. As we see in the cartoon, sometimes that termination sequence will make a hairpin. Okay, sometimes there'll be proteins that bind to it. All right, so we have various things that can happen here. Questions on RNA polymerase? What's going on there? Okay. Then I want to talk about regulation of transcription. And we saw one of these in terms of what you did in lab. One of these was effective, but there are there's 4 different ways this can happen. Okay. We have some more terminology that I want to introduce. Okay, so we're going to talk about an operator. An operator is a DNA sequence that is bound by a repressor, which is a protein. Okay. The opposite of an operator is an enhancer. Is a DNA sequence that is bound by an activator, which is also going to be a protein. Questions on that? What's a protein? What's a DNA here in terms of the terminology? So in terms of, you need to be able to define operator, repressor. Repressor and activator are pretty good, right? They kind of know what they are. Enhancer too. Operator is kind of less of a word or a term. The word tells you what it does. All right, there are questions? And there are 4 kind of ways we can think about this regulation happening. So we have our negative regulation here, where our operator is going to bind. It's not going to be there for, you're generally going to have both of these in concert, some mix of them. Okay, so I guess we could mix it 4 different ways, right? This with this, this with this, this with this, this with this. But we've got an operator here that is bound to the promoter, inhibiting. And then when a special molecule, when a signal molecule comes along, it will bind to that operator and or bind to the repressor, and the repressor will fall off the operator. Let's go ahead and write that down. So I'm going to write these below. So 4 ways that the initiation of transcription can be regulated. All right, so, but they're not separate from each other. We're going to have, they could be separate, but we're often going to have both an operator and an enhancer active for a gene. And in fact, that was what was going on in lab, although you only manipulated the operator part. So 4 ways we've got the operator is bound by a repressor that, the repressor, blocks RNA polymerase from binding. Okay. When that repressor is bound by a signal molecule, it changes shape and it falls off the operator. So it falls off the operator and then RNA polymerase can bind. All right, and in fact, we'll talk about it in detail below, but this is what you did in lab. You had the lac operon. Look at this. You had the lac operon in front of your MTHFS gene. The lac repressor was bound to it. You added IPTG. You added that, you let it go overnight, and the IPTG came in and bound to the repressor changed shape and it fell off, and then this was open for RNA polymerase to bind. Okay. That's what you see over here, except we won't worry about the enhancer. We could do the enhancer next, or let's do the other form of what happens. So the other form is similar. It's just when the signal molecule's bound to the repressor is bound to the operator. Okay, instead of the reverse. Does that make sense? You see both of these. So 2 would be when the repressor is bound by a signal molecule. It binds to the operator. And prevents RNA polymerase from binding. Okay. When that signal molecule is removed, the repressor will change shape and fall off the operator. This is not, and then RNA polymerase can bind. I'll just leave that out. You can add it if you want. That was not what you did in lab. Okay. You added a signal molecule. The reason why you added IPTG, and you'll see this in the instructions for the lab report, and by the way, I hope you've noticed that for the lab reports, the answers to all the questions are in the introductions to every lab. So you just need to read through that and you'll find those answers. But you added something. The reason why you used IPTG instead of lactose, which is what the repressor evolved for, is because the cell eats lactose. So you add IPTG, it can't consume the IPTG, so it's going to always keep that repressor off, if that makes sense. Otherwise, you'd have to keep adding lactose back. You guys are probably sick of how much time you had to wait for things to happen in a lab already. Okay, positive regulation. So those were two examples of positive regulation. The reverse here, not reverse, but for positive regulation, and this is also going on in your cell, in the lac operon, all right, is that not only can the RNA, does the RNA polymerase need something not in the way, but it needs something next to it to help it bind. Okay, and that's where we have one of these enhancers, or we have an activator bound to an enhancer for that to happen. And we'll talk about the specific one that was going on in lab, but you didn't need to worry about this because this was, whether it was bound or not, depended on glucose and cyclic AMP being made. That's down there. We'll look at that in a minute. Okay. But your cells had eaten all the glucose by the time you got 7 hours out and added your IPTG. Yes?
Did you say the first two are positive?
The first two are negative regulation. Let's do this. Okay, and then now we'll talk about positive regulation. Okay, so for positive regulation, I guess I could start at 1 again, or I'll just do 3 because that's what it wants to do. But for positive regulation, what we've got on here, going on here is we have this activator, okay? So an activator binds to, or is bound to the enhancer. It's bound to the enhancer when, let's look what's going on in our picture for our first one here. Is it when the signal molecule is bound or not when it's not bound? Or when, so top one, when it's not bound, right? Okay, so an activator is bound to the enhancer when no signal molecule is present. When that signal molecule is made, like added could be you added it to solution, it binds to the activator and the activator falls off the enhancer. All right. The opposite, and then the RNA polymerase can only bind when the enhancer is bound. Let's do this. Regulation up here. The RNA polymerase can only bind when the enhancers, or when the activator is bound. This is a place that during the pandemic, I decided that I was going to, when we were doing our courses online, I decided I was going to look up videos online that were good. I don't know if I've told you the story before, like my dad. And I spent so much more time trying to find videos than it would be to just record the lecture. So I decided this is the wrong way to do it. But this was something where I got in a Khan video. Usually I really like the Khan Academy videos, so I recommend them for things. But they were like, yeah, when the enhancer's there, you get a little bit more. No, it's really all or none, almost. Might be a little bit leaky, but it's not good much. So an activator, it was being given by Khan, who's a mathematician. And you know, He's a far better mathematician than I am well, maybe we're at the same level for biochemists, but when we swap, he's a better biochemist than I am a mathematician. Let's say it that way. So I'm critical of what he did, but I can't speak out that much. Okay, so when a signal molecule is added, the other one is when a signal molecule is added. The activator changes shape and binds. Not the repressor, the enhancer. And RNA polymerase can bind. Okay, so we got those four different, and this is what actually happens in the lac operon, this fourth one. So you're really looking at 1 and 4. Okay, but you didn't need to worry about 4. 4 was about whether there was glucose around. Low glucose results in cyclic AMP being made. All right, so we'll go ahead and we'll look at that here real quick, and then I'll hand the exams back. All right, so we're going to apply these here in terms of lac operon. So what we have is we have transcription occurs here. Okay, and this is no transcription because you don't see the RNA polymerase bind. And so we have a lac repressor that's bound. And when lactose is added, what happens is that lac repressor, the lactose binds to the lac repressor, this thing, and the lac repressor falls off. Okay. So we could say lactose binds. And the reason for this in nature is that if you're a bacteria and there's no lactose around, this will be bound, right? And if there's no lactose around, you don't want to waste energy making proteins that help you digest lactose. So what's the other part of this? So if we have high cyclic AMP when we have low glucose. Well, it turns out that glucose is easier to metabolize than lactose. You need kind of all the same genes. All right, when we think about this. But then that's going to get shoved into glycolysis, right? The lactose can be shoved into glycolysis. So if there's glucose around, if there's a lot of glucose around, you don't want to be making lactose genes. You want to eat the glucose first and then eat the lactose next because lactose costs more. You don't get as much energy out of it. Yes?
So you're saying like when lactose is present, it'll bind to the lac repressor to so that it's allowed to process.
And the lac repressor will fall off.
So that the protein can be made to process the lactose?
Yes, when there's lactose around, the lac repressor will fall off so you can make lactose. So you can make the proteins that process lactose. Okay. There are 3 specific enzymes that are involved here. We're not going to go into them in detail. You know how this works, especially because we used it in lab. Same thing's happening here. Lactose gets added, right? The lac repressor gets bound, it falls off, but no activator gets bound here because we have high glucose around. The bacteria want to eat up all the glucose and then they'll start on the lactose. Okay. Remember, as a bacterium, you're competing with all the other bacterium around you and you want to grow and divide as fast as possible, take over the culture first. Okay. So you're going to prefer that. And so this is what you were using in lab. You added IPTG, which is a lactose mimic. Okay? And so you caused this to fall off. And in your case, cyclic AMP was being made because you did it 7 hours in, right? And those bacteria had already eaten all the glucose. Questions? Yeah.
you're lactose intolerant, is it a problem with this process? Or?
This is bacterial. So in terms of being lactose intolerant is you are not making lactase. You may know this. Do you know this already? Should I not bother? Okay. You're not making
Sure, anything to it. Like, is it part of the genome? Or is
Lac-
it part of the, yeah.
lactose for you.
Yeah.
The lactase for you is part of your genome. And it's supposed to turn off after you stop nursing so you don't waste energy making enzymes to break down milk. Because if you go back to caveman days, we didn't have cows, right? And so the difference now is there's a bunch of dairy and cows and things like that. So if your ancestors are European, you're more likely to not be lactose intolerant because cows are common there. If your ancestors are Asian, you're more likely to be lactose intolerant because there weren't a lot of cows there. And so milk wasn't available. But Europe was an advantage to be able to digest its own food.
I'm severely lactose.
Once you have yours, you're welcome to take off. I think I posted the answer key. If I haven't, don't post the answer key. I'll do it up right after class. I'm also happy to answer questions right now.
I crammed this.
Well, I think it was like 10 days later, and so I was kind of still in my like despairing mode. But for some reason, my brain was like,
Oh, I knew I was.
All right, see you guys all soon in lab.
oh yes.
It's gonna be fun. It's kinetics.
Yeah, except we have to do it all ourselves.
So you guys usually work in groups anyway, right?
Like, we have to make up the
Yeah, you're doing it on your own, right? You're doing your own thing,
But it says like we have our makeup procedure.
yes.
Yeah, that's what I
It's
got.
fun stuff, isn't it?
Yeah, it really is.
Okay.
Yeah.
If you're not having a coaching session
Yeah.
Thursday,
This Thursday, yes.
what? Oh, I guess we have lab today.
We do. Do you want to talk about stuff for lab report? Do you want to talk about stuff in general? You want to talk about?
I have a question just because every single time, well, the first time we did an ATP question, I didn't get to it, and this time I actually was able to do it.